come to the HeLa cells from a totally different point of view. When I arrived at UCSB in about 1978 as a postdoctoral fellow in biology, I joined a lab that was and still is studying cancer drugs, and I took on the project of trying to understand how one of a very prominent, actually then used class of drugs called the vinca alkaloids, vinblastine and vincristine, how it actually worked, what its mechanism was. They were known to work, but we didn't know how they worked. And when we decided to start the study, we wanted some cultured cells to use, and the most prominent, most well-known culture was HeLa. I had no idea where it came from. We would order it from the American Type Tissue Culture Collection, which is a nonprofit organization that provides cultured cells to researchers for a price, but the price of carrying them, because as you carry them over months or years, they change. So if you want to go back to the original that you started with, you go back to ATCC, American Type Tissue Collection, and get some that haven't been growing, that have been frozen away. So we used those cells for many years, Professor Leslie Wilson and I. Now we use many more kinds of cultured human cancer cells, specifically from prostate or breast or lung, and rarely use HeLa, but we probably did maybe 20 or 30 studies on HeLa, and I have them here if you'd like to just look at them. They aren't particularly fantastic looking in this microscope, but I also have some images I pulled out of them that I can pass around. This is an image of the surface view of HeLa cells at about 2,000 times magnification, and this is just a human chromosome image. And then these are some images we actually took of HeLa cells many years ago when we were studying the vincristine and vinblastine and some more experimental drugs. And I've outlined a dividing cell for you, and this is without drug, and you see the chromosomes in blue and the microtubules, which is the target of the drugs we study, in yellow. And then in one, I've chosen 3 of the drugs, and you could, sure. Oh yeah, and so you see a very orderly, if you're familiar with biology, of mitosis with the chromosomes lining up in the middle, and here they're dividing into 2 new cells. And then in the presence of the drug, all of this orderly arrangement gets disordered and screwed up, and the cells block in mitosis, and after a few days they die. And so I've included a couple others that are like that, but instead of seeing this orderly chromosome plate in the middle and the microtubules pulling the chromosomes into 2 new cells, you see the chromosomes are all around the central microtubules, and there's nothing to do, to actually carry it out. And so we've studied a number of cancer drugs, both with National Institutes of Health funding. I get no funding from the University of, well, not none, but almost 0 from the University of California. All of my salary comes either from National Institutes of Health or from drug companies that pay my salary to collaborate with them to figure out how their drugs are working, because they often find a drug is working and it goes into people, but they don't know how it's working. And so they come to us because we're experts in this and to find out how they work. And originally HeLa cells were our most common and wonderful cell line. The fellow who just brought them up for me, the technician, graduate student actually, who came over here with the microscope, said, these have been frozen down 5 years, and 4 weeks ago I unfroze them, and they're growing like gangbusters. Wow. So it's really a phenomenon. So I think that's, and just to add a little bit to what you've said, I was amazed to find out that the story of Henrietta Lacks. I had taught cancer courses at UCSB in the 80s and 90s, and And I knew something of her history, but I had no idea of her personal history. And then the other thing is that I do know that, as you say, it's disadvantaged people or people who don't have knowledge, or people, poor people or black people, who tend to be were especially then taken advantage of. I don't know if that's as much the case now or not. But I also know that throughout history, certainly non-black people have been taken advantage of. Edward Jenner, when he was developing the smallpox vaccine, took a young boy and exposed him gave him the cowpox, which was what he thought was a source of antibodies to smallpox, and then sent him through the smallpox wards to see if he would get smallpox, and he didn't. So there certainly must be examples of misuse. And this was in England. I could have been black, but I doubt it at that time.
But in terms of the systematic,
right?
I would the way in which it just happens. There's a pattern. There's this consistency we can see. And like I said, as Dorothy Roberts outlines in Killing the Black Body, these things are still happening today. Yeah, unfortunately.
And I think they probably happen to many of us. It takes so much knowledge to be able to ask the right questions when you're in the face of an MD. So I'll pass these around. And